Every surface we touch is teeming with microscopic life. We live constantly surrounded by bacteria, viruses, and fungi, organisms collectively known as pathogens. If these microscopic invaders manage to enter a human host, they have one goal: to multiply. When they do, they can disrupt normal body functions and cause transmissible diseases. Take a disease like cholera. The cholera bacteria attaches to the wall of the small intestine and secretes a toxin. This toxin forces the intestine lining to release chloride ions, which lowers the water potential in the gut. Water from the surrounding cells then floods into the intestine via osmosis, leading to severe dehydration. Because these invisible threats are constant and potentially deadly, human survival relies on executing a highly coordinated, multi-layered biological defense playbook. The first play in this defensive system is a mechanical barrier. The human skin acts as an impenetrable physical wall against infection. But we still have to breathe. To protect our airways, the body uses secondary mechanical traps. Hairs inside the nose act as a natural filter, while a layer of sticky mucus in the respiratory tract captures airborne pathogens before they can reach the lungs. If a pathogen sneaks in through the food we eat or the water we drink, it hits a chemical barrier. The stomach produces concentrated hydrochloric acid, which is strong enough to destroy most microbes on contact. Humans have also engineered massive external defenses to complement our biology. This historical photograph shows workers building large brick sewer pipes, part of the sanitation and sewage treatment systems designed to remove pathogens from our environment before they ever reach our physical bodies. These combined physical and chemical barriers are highly effective, but any tiny breach, like a scrape on the knee, requires a highly specialized internal rapid response team to prevent an infection from taking over. The moment a pathogen breaches the physical barriers and enters the blood, a specialized team of white blood cells deploys. Looking at this photomicrograph of human blood, the first responder is clearly visible. This single white blood cell on the left, characterized by its distinctive lobed nucleus, is called a phagocyte. When a phagocyte encounters a germ, it initiates a process called phagocytosis. The cell extends its membrane to completely surround and engulf the invading pathogen, pulling it inwards. Once trapped inside, the phagocyte releases digestive enzymes, breaking the invader down into harmless particles. Phagocytosis is a fast and brutal defense, but it is nonspecific. Phagocytes will attack anything foreign. To defeat rapidly multiplying threats, the body requires a more strategic, targeted backup. Returning to our blood sample, we can see the second major type of white blood cell. On the right, these darker cells with large, round nuclei are lymphocytes. To understand how lymphocytes work, we have to look at the pathogens themselves. Every pathogen has uniquely shaped chemical markers protruding from its outer cell membrane. These specific markers are called antigens. Lymphocytes read them and manufacture specialized proteins called antibodies. They create antibodies with a perfectly complementary shape, allowing them to physically lock precisely onto the pathogen's antigens. Once attached, these antibodies incapacitate the pathogens by causing them to stick or clump together. This restricts their movement and marks them for mass destruction by the phagocytes. After the infection is defeated, the lymphocytes leave behind memory cells that continue to circulate in the blood. These memory cells provide what is known as active immunity. If that exact same pathogen ever returns, the memory cells instantly mass-produce the correct antibodies, stopping the disease before it can take hold a second time. We can contrast this lifelong protection with passive immunity, such as a baby receiving antibodies directly through its mother's breast milk. This provides immediate, critical defense while the infant's immune system develops. But because it doesn't create memory cells, the protection is only short-term. There is a central problem with active immunity. Earning that lifelong protection requires surviving an infection from a live, dangerous pathogen. As this historical photograph of a crowded hospital ward illustrates, that biological battle can lead to severe illness or death. Medical science found a way around this risk: the vaccine. A vaccine safely introduces a weakened version of a pathogen, or sometimes just its harmless antigens, directly into the body. Because the pathogen is weakened, it cannot cause illness, but the antigens trick the lymphocytes into launching a full immune response, producing the specific antibodies and, crucially, those long-lasting memory cells. Vaccination acts as the ultimate practice drill. It safely grants the host the lifelong protection of active immunity without the host ever having to suffer the actual disease. The impact of vaccines extends far beyond the individual. As seen in this historical photo of a crowd lining up for polio shots, mass vaccination directly limits where a pathogen can survive. This creates a protective shield known as herd immunity. In a highly vaccinated community, transmission lines spread quickly among unprotected individuals, but they stop dead when they encounter a thick cluster of immune hosts. When the vast majority of a population is vaccinated, the pathogen simply runs out of vulnerable hosts to infect and breed within, entirely breaking the chain of transmission. From the mechanical barriers of the skin to the heavy tackling phagocytes and strategic lymphocytes in our blood, all the way to the community shield of herd immunity, the human body and modern medicine work in close coordination to keep us alive.